So imagine you arrive to a city of a country you don't speak the language, and you're trying to figure out what people do for a living. If you see a school and there are a lot of kids in the playground and an adult, it might be really obvious that person's job is probably a teacher. But maybe there's just a parent picking up their kid. Instead, if one day all the schools are closed and you see a bunch of people protesting in the streets, now it's very obvious that those are the teachers and their work is to run the schools. In the same way, we can study proteins. Cells are actually very similar to cities. As our cities, they're also compartmentalized and divide the task in different places. So there are compartments dedicated for decision-making, others for energy supply, and the workers of these microscopic cities are the protein. If they go on strike, also our mini cells suffer. I'm Rosario Valenti, and I make proteins go on strike to understand what's the function in the cell. A lot of our disease come from problems on how the proteins are working. So understanding the specific role of each of the proteins would let us figure out how to treat different diseases. So the way I make proteins go on strike is by using what's called the auxin-induced degron system. In this system, we twitch the genome of the cells so that our protein of interest it's made with a small tag on it. And also we make it with a green fluorescent protein or GFP so that we can visualize the whole process. We also twitch the cells to express the protein tier 1, an adapter protein. This tier 1 protein won't do anything to our protein of interest until we decide to trigger this mechanism. For that, we add to the cells the small molecule auxin, and this auxin brings these two parts together and marks the protein for rapid degradation. So this means that in the cell, the protein that I'm interested in is working and doing its job as always, until I decide to trigger the experiment. So when I want to make the experiment, I add the auxin to the media, and that depletes the protein in 30 minutes, and I can see what immediate response the cell has. Now, though each city is unique, all cities share some commonalities. In the same way, all the cells share fundamental processes of cellular biology. This means that we can use yeast cells that are much easier to work with and yet learn things informative also for human biology. Specifically for the baker's yeast, Saccharomyces cerevisiae, there are over 5,000 different proteins in each cell. We implemented the AID system, one protein at a time, for all of them. So now it's like a comics multiverse. In each of our microscopic cities, we are removing one player, and we can see in all of them, side by side, what happened. For this, we use a robotic system such as this one. We believe that understanding these dynamic aspects of the cell would give us fundamental and deeper understanding of how the cell functions.